Hi, everybody. Good morning. I'm Burcu Tepekule. So I'm going to be an incoming assistant professor, BRCCH professor, affiliated with the University of Basel. And today, I'm still assembling my group. So today I will talk about some of my work from my postdocs in the context of pediatric health. So my job is to do mathematical modeling. So before I delve into results, I want to talk about a little bit about mathematical modeling is, and then what it actually isn't. So there are multiple types of questions one can answer with mathematical modeling, and we can group them into these non-mutually exclusive categories. So we either want to merely predict the quantity of something, like maybe the number of infecteds in a month from now in a pandemic, or we want to understand mechanistically what drives certain dynamics in the system, or we want to intervene, which means we want to design hypothetical therapeutic interventions that might have the potential of being clinically relevant. And most of the time, these questions cannot be answered with one methodology alone. And we have this distinction of black and the white box models. So black box models are like these classical machine learning algorithms or more advanced neural network techniques, where you have the power of prediction when you have a lot of data. But of course, the box is black, so you really do not know how the prediction happens. So it gets a little bit tricky to interpret the outcome. And on the other hand, we have the white box models, where we can build systems where we put the mechanisms in. So it becomes a little bit easier to interpret the outcome, but it's of course not guaranteed. And these kind of models are great for testing novel intervention strategies that you never had the data for, or running counterfactual scenarios where you can never observe in real life, and also generating huge amount of data. Then you can feed back into the black box models and help some prediction. So in the end, it's all about going back and forth between these two approaches. And as an example, these are the methods along this gradient for modeling the microbial communities. So you start with data analysis, and then as you move down in this gradient, each step feeds the upcoming one. And ultimately, you end up with mechanistic models, which are the models I will be focusing on today. But I want to make one thing clear. Mechanistic modeling is an abstraction. So in fact, most of the time, our aim is to make it as simple as possible with keeping the mechanisms relevant to the scientific question at hand. And honestly, the math is the easier part of it. So most of the time, our job is to review all the possible mechanisms in the literature, synthesize them, and systematically introduce or remove them to make the most relevant model. And these models are also very helpful tools for simulating completely unrealistic scenarios, which helps us identify actually what drives the outcomes in reality. So the goal is not to replicate the wet lab experiments or cohort studies verbatim, but to have the outputs of these studies as like a subset of the model outputs. So with this, I will now move onto the mechanistic models I worked on during my postdoctoral work. So as a general theme, my work is concerned with how we learn to live with our microbes while we're growing up, and the consequences of failing to do so. And we use multiple approaches, including statistical modeling of human and animal data. But today I will focus on two projects that are based on within and between host modeling that model the reciprocal ontogeny of microbiome and immune maturation. Okay, so the question how we learn to exist with our microbes is super relevant in the context of pediatric health. Right? Because when infants fail to maintain a homeostatic relationship with their developing microbiome, the downstream effects are pretty serious. So we know from a vast body of evidence that early life dysbiosis is associated with conditions ranging from childhood obesity to asthma, allergies, neurodevelopmental disorders, increased susceptibility to infectious diseases, etc. And these downstream effects are primarily mediated by the immune response. So the children's immune system governs how they react to infection and vaccination, but at the same time, it significantly impacts their metabolic and cognitive development. And this relationship between the microbiome and the immune system is a bidirectional one. So the immune system shapes which microbes can persist, and the microbes in turn shape the immune system. So that is why, although the dysbiosis might start locally at the mucosal sites, it has systemic consequences. And there is an added layer of complexity when we study children, of course, because children grow, right? And in this context, growth happens within a critical developmental window, roughly the first 2 years of life, during which the immune system is especially receptive to environmental influences. And once this window closes, the changes become largely permanent, or in other words, they get imprinted. So we were interested in developing a mechanistic model where we could formalize the relationship between the developing microbiome and the developing immune response within this critical window. And since the scientific question is how we learn to live with our microbes, we were focusing on the adaptive arm of the immune system, specifically the IgA responses in the gut during the first 2 years of life. Okay, so what does our model entail? So this is a schematic of the model that I will walk you through. So the mathematical model we built included the nutritional and microbial inputs from breast milk and complementary feeding, the influx of maternal antibodies, an ecological model of the gut microbiome that integrates the role of antibodies in shaping the community composition, the function of the gut community in regulating the inflammatory tone of the microenvironment, the role of gut epithelium in transporting the bacterial antigens into the underlying mesenteric lymph nodes, and these antigens activating B cells and contributing to the balance between follicular T regulatory and T helper cells, and the dynamics of affinity maturation and plasma cell generation, and finally, the control of the microbial composition by endogenously produced antibodies, closing this loop of reciprocal interactions. So this illustration represents a big system of delayed differential equations, and was parameterized by using Bayesian inference techniques and a combination of multiple data sets, including 16S RNA sequencing, flow cytometry, and IgA sequencing data. So this is how the output of the control scenario looks like. So first, based on multiple sources of data, we first model inputs of feeding durations and the concentration of HMOs, calories, maternal antibodies transferred to the infant through different modes of feeding. And then we fit our model on the 16S RNA sequencing data, flow cytometry, and IgA sequencing data simultaneously. So one key assumption during model fitting here is that under the average feeding pattern in the control cohort, the infant develops an antibody profile that resembles the nursing mother's. So this establishes a transgenerational cycle of immune imprinting. So this assumption is a strong one, but it's grounded in observational data. And fitting on this data gives us the trajectory of the inflammation in the gut lumen and the affinity maturation of the endogenous antibodies over time. So another key modeling choice here is how we map antibody affinity to function. So we assume low affinity IgA predominantly masks symbiotic commensals. It promotes their residency in the gut, while high affinity IgA predominantly neutralizes and expels bacteria from the gut. And what we want in healthy development is a masking response to symbiotic commensals and a neutralizing response to pathogens. So a learning process here that we're interested in is this within host evolution of these antibody responses. And this process depends both on which form the bacteria are sampled to the mesenteric lymph nodes, plus their intrinsic inflammatory capacities. So you can think of it as like bacteria can be sampled in an IgA-bound and IgA-free form. And IgA-bound bacteria are like pre-tagged by the maternal antibodies, which bias the entire process to the tolerogenic profile. So this tagging is exactly where maternal antibodies can signal to the offspring, hey, these are commensal microbes, so you shouldn't treat them as pathogens. And in the case of pathogenic microbes, IgA will be high affinity anyway. So the ones that are actually IgA-bound will be expelled from the gut, and they're not going to be sampled to the mesenteric lymph nodes, and the ones will be IgA-free. So they're not going to have this tolerogenic programming by default. And their inherent properties are going to be more inflammatory anyway, so we will more likely to see a neutralizing response. And of course, this is a simplification. IgA biology is way more complicated than that, but it allows us to model a continuum of responses that emerge from shared bacterial traits and common principles of antibody production, rather than just hard coding what kind of response we should have for what kind of taxonomic group. Okay, so using this model, we then address 3 questions that we think are highly relevant in the field. So first, we focused on the earliest form of malnutrition, which is insufficient breastfeeding. And this is actually a real concern in low-resource settings because most mothers have to go back to work relatively quickly after giving birth. And lack of breast milk means many things. It is lack of nutrition, it is lack of protection against infections, but it's also lack of this transgenerational immune priming that I just talked about. So it's just lack of also information regarding which microbes are supposed to be symbiotic commensals. So as an example, here you see the model output where the mother exclusively breastfeeds for 15 days and continues with mixed feeding for 15 more days before cutting any breast milk at all. And you can see that our model predicts a hyperreactive antibody response against the symbiotic commensals. So basically, the endogenously produced antibodies do not mask the commensals anymore, but they target them as if they were a threat, as if they were like pathogens. And this hyperreactive response is accompanied by a persistent inflammation in the gut. So we saw that when we compared the babies from Zimbabwe cohort and the burn birth cohort, and we can see that the Zimbabwe cohort has like a persistent inflammation, where the burn birth cohort actually has a decreasing inflammation. So this is only one example. The beauty of having a mathematical model is like we can have thousands of scenarios that we can simulate. So we can systematically simulate different scenarios to compare them to the control. For example, in all scenarios where there was no transfer of breast milk, we observed the maturation of a hyperreactive response in the infant. So we couldn't rescue the pathological immune printing by just like giving probiotics. But interestingly, in terms of the transgenerational transmission of IgA profiles, our model demonstrated a modulation of hyperreactivity against symbiotic commensals rather than the complete transfer of it. And actually, if you think about it, this is great news, right? So because over multiple generations, this means that this reactivity in the mother's antibodies will be diluted and vanish over time, which also makes sense from an evolutionary perspective. And it also means, at least within this context, it is safer to breastfeed than not, regardless of the mother's antibody profile. And the mechanistic interpretation of this is that antigen-agnostic properties of IgA, meaning that its ability to promote M cell-mediating antigen sampling or program dendritic cells toward tolerogenic profiles, they remain functional even if the IgA itself or the antibody itself is like hyperreactive. And coming back to probiotics, so they didn't rescue the immune response, but they substantially reduced the pathogen burden in the gut lumen during the first 30 days of life, which is super important actually if you want to avoid enteric infections, which can be fatal. So the ecological benefit of colonization resistance is not to be underestimated here. The next question we were interested in was whether we can identify markers for early prediction of pathological imprinting. To answer this question, we quantified the predictive power of Enterobacteriaceae abundance in stool samples at different time points during development. And we asked how well a single fecal sample can predict whether the infant will develop a tolerant or a hyperreactive immune profile by the end of 2 years. So this plot, it's here a predictive power of 1 means discrimination between pathological and healthy trajectories for sure, and a 0.5 basically corresponds to a random draw. And the first thing we observe is that the total abundance of Enterobacteriaceae is not very informative during the first 6 months of life. This is because there is two compensatory mechanisms that are regulating its abundance simultaneously, right? So one is ecological competition, the competition with commensals that is regulating its abundance. And the other one is actually activation of the endogenous immune system in case the maternal antibodies are insufficient. But in contrast, IgA-bound Enterobacteriaceae abundance during very early life is very highly predictive because it directly reflects the sufficiency of the maternal antibodies. So if the mother's IgA is not neutralizing Enterobacteriaceae enough, you will see less IgA-bound Enterobacteriaceae in the stool, which already predicts a hyperreactive response. But then something interesting happens as we approach month 6. So the predictive power of IgA-bound abundance drops, while the total abundance becomes more informative. And this is actually the transition period from maternal to endogenous antibodies. And actually, during this window, the IgA-bound signal is not very informative because it's kind of a mix between like the maternal antibodies and endogenous antibodies, so it's hard to interpret. So in the end, the combination of when you collect the fecal samples and what you measure in them determines what you can learn. And this is directly relevant for clinical study design, especially when the resources are limited and you can only sample at certain time points. So the last question we focused on was, how very early postnatal or perinatal events can impact later stages of ontogeny. So it's actually a very interesting question because most of the time, immune cells that are ultimately affected by very early postnatal events are not even present yet when these signals are received. So to address this question, we investigated the effect of pre-weaning events on the immune response against post-weaning commensals. So these commensals are only introduced after the weaning time, meaning that like after mixed feeding. And then they're not present during the exclusive breastfeeding period, which means the immune system does not actually see them. And despite this temporal succession, our predictor importance analysis revealed something very counterintuitive. It actually showed that the exclusive breastfeeding duration has a substantial effect on the affinity maturation against post-weaning commensals. And looking deeper, we saw that a significant portion of the information is carried through the inflammatory tone of the microenvironment, whereas the details of the ecological composition was less influential. And this finding is very important because it quantifies how much the functional outcome of ecology matters compared to which taxonomic groups or species are in there. And this insight allows us to go one step further than managing the gut ecology and actually control the inflammatory response directly. So in fact, we checked whether TLR4 antagonists could help. And our model showed that even when the pathogenic burden is high, their administration can help imprinting of a tolerogenic response. And of course, breast milk does this naturally through HMOs, right? So this analysis shows us, even with the very basic feedback loops we put in this model, we can have at least one mechanistic explanation as to why breastfeeding continues to shape immune responses long after its cessation. And I think the next question that naturally follows from here is whether the process of immune education during the first 2 years of life is actually a chaotic system, meaning that the very small differences at the initial conditions can change the trajectory of the system entirely. And we have both data for it and data against it. So we see a lot of studies that associate very strongly certain outcomes with delivery mode, for example. But on the other hand, not every baby that was not breastfed is developing a disorder. So I think these competing forces, whether the system is chaotic versus there is certain level of resilience in the system, is actually a very fascinating one. And of course, this is not only a mathematical exercise, right? Because you want to know whether any intervention at a given time point can really push this trajectory that goes to a pathological attractor to go to a healthy attractor. So we really would like to understand that. So this project was about our within-host model. And here we show that different types of disruptions to critical window can have lasting microbial and immunological consequences. So now I want to take one step back and look at this phenomena from more like an evolutionary biology perspective. So this window has likely evolved because it enables the host immune system to learn to tolerate commensal microbes that are likely to encounter later in life. So this window is supposed to be a representation or like a sneak peek that is like about to come later in life. But this definition, of course, raises also more questions, evolutionary questions actually, like how long should this window be, or what kind of factors shape this window length? Because there are both pros and cons to keep this window short and long. So if you keep it too short, you might not have enough time to encounter the commensals that you need to encounter. But if you keep it too long, your chance of having a fatal infection increases. And also, you're just also paying the price of basically keeping it open and being vulnerable to any kind of threat overall. And to explore this question, we built a between-host model, or an epidemiological model, where individuals move through epidemiological states. So like susceptible, colonized with commensals, infected by a pathogen, or both. And after this critical time point, the critical window ends, they move to either like a tolerized or a non-tolerized state. And then we incorporated multiple factors that we think might be shaping the optimal window length. For example, between-host transmission rates, vertical transmission of microbes, duration of infection, and host demography, because all of these will actually determine how much and when you're going to be exposed to microbes. And then we play around with these rates, and for each scenario, we estimate the fitness associated with all the possible lengths of the window. Then we find the length that corresponds to the highest reproduction rate, meaning that it corresponds to the highest fitness. And that length is the optimal window length. And by doing this systematically, we can look at which factors would lengthen or shorten this optimal window. So one of the interesting results is that the optimal window length is not always monotonic with respect to bacterial transmission rates. So here on the left, as the pathogen transmission increases, the optimal window length drops sharply. And it makes sense, right? Because the cost of keeping the window open while pathogens are circulating very fast, it just becomes too high, so you kind of better close it. But on the right, we see the effect of increasing the rate of commensal transmission. And here the pattern gets a little bit more interesting, because when commensal transmission is very low, the optimal window length is actually 0. So there is no point keeping it open, because all you do is paying the cost of keeping it open. But once the commensal transmission rate crosses a threshold, the window jumps open, and then it again decreases, because now your chance increases of encountering a commensal, and you can get by basically with a shorter window. So the optimal strategy depends on the microbial ecology. But the host demography also matters. So as fertility increases, the average age of infection goes down, because basically you have more susceptibles that could spread the infection or spread the commensals, which drives faster transmission and earlier encounters. So this enables a shorter critical window because hosts acquire tolerance earlier. And similarly, if fertility is low, you need longer critical window because microbial encounters happen later in life. But low fertility can be compensated by vertical transmission of microbes. So if the mother passes commensals directly to the offspring, the host gets exposed really early regardless of the population level transmission, which shortens the window. And what does this results really mean for us today? I mean, industrialization did not select our window length, right? For our window length was selected probably in an environment where we had way more transmission. So probably our windows are too short for the conditions we experience today. But our model also points to a potential buffer, which is the vertical transmission of commensals. So we already know that the birth mode, skin-to-skin contact, and in general, parental microbiome transfer is very important in industrial settings. But one of the reasons for that is because we cannot simply afford to wait it out. Like vertical transmission shortens the required window by guaranteeing early exposure, so we have to hasten this process by making use of it. And now one direction we're planning to work on is embedding our within host models into age-structured epidemiological models to quantify how disease spread is influenced by a suboptimal immune response. And this integration is important because in the end, epidemiological models are only as good as the assumptions you make about individual compartments, right? So without incorporating the immune ontogeny and age structures, we are likely to miss many aspects of disease transmission, like susceptibility, infectiousness, vaccine efficacy, treatment success, even when modeling mixed populations that include adults. Anyway, needless to say, mathematical modeling is an interdisciplinary effort more than anything. So I want to thank all my theoretical, experimental, and medical collaborators who grounded these projects in their expertise. And thank you very much for your attention. If you have any questions, I can take them now.